Hi everyone, I'm Martin Battle. I'm a postdoctoral research assistant at the University of Essex, and today I'm going to talk to you a little bit about my time in Tigris. So I've been working in FP2A, looking at sorghum and pearl millet, and today this talk is a whole plant perspective crop improvement. But as well as that, I'm obviously going to be talking to you a bit about Tigris and what Tigris has meant to me and some of the biggest successes for me personally in Tigris. So some of you might have already heard a talk from my PI, which is Professor Tracy Lawson at Essex. If you haven't already heard her talk, please go and listen to that one as well. It's in this same workshop. But Sheila told you a bit about how our lab's been collaborating as part of Tigris and the connections that we've made as a lab. So I'm going to talk to you more instead about the things that personally have meant a lot to me through Tigris and the ways in which it's really helped me personally. So it's a little bit of a different perspective. And then I'll give you a quick glimpse into some of my research, which won't go into too much detail, and some of the things that we're looking to publish soon. So without further ado. So for me, Tigris began in late 2018. I believe I began on November, very early November in 2018. And when I started Tigris, I was a research technician under Sylvain Valet-Chavron, who's now gone on to run his own lab elsewhere. But for me, Tigris was always this big, international, exciting, research commitment. It was always something that was way beyond the scope of anything I'd ever worked on. I was always just used to working in one little lab. I came from a lab where I was the second ever PhD student. We had no postdocs, and I come into a lab with Tracy Lawson having a far bigger lab than what I was used to, with far more equipment, and being very excited to be able to work there. But Tigris being a step way beyond this, it was a huge project on the scope of things that I didn't know I'd be able to be involved in. So that was already hugely exciting to me. And then when I stepped in, February 2019, we're planning for this event not long before that, but I've not started Tigris for long before we start talking about, okay, when are you going to go out to India? And to me, that's amazing. I mean, if you told me that for one of my first projects post finishing my PhD, I'd be able to be traveling to India to work with all of these different people that otherwise I'd never have interacted with at all, and that it would happen immediately as soon as the project started, I don't know what I'd say. But this workshop for me was where Tigris really kicked off. And I'm not sure whether, Dr. Gitanjali Yadav, who set this up at NIPGR, I'm not sure if she realizes how impactful this was for me personally, but I really want to thank her for this. So this was just a bioinformatics workshop in ours. So we were spending a few days, or I think it was most of a week, in New Delhi with lots and lots of people from NIPGR and across India who had come together to do this training workshop. And on the surface, that was really great to begin with. But what took this up a step for me beyond the training, which I have continued to use and has been really invaluable through my research, and I've built upon all of that training since then. But what really made this event so memorable for me was that it coincidentally happened at the same time as a meeting between FP1 and FP6 in New Delhi. So for me, going out thinking, okay, I'm going to get to learn some stuff, and I might meet a few people, and I'll meet the people from my own flagship project. But I also ended up getting to meet so many more people. And this was really my first opportunity to see Tigris as the community that it's become over time, and the first glimpse into what Tigris really could be. So this for me was a big highlight of early Tigris. And then my second trip out to India was the General Assembly in January of 2020. And this event was also fantastic. So we had people from across all the flagship projects, and I'm sure plenty of people have talked about this already. But this event really allowed Tigris to feel, again, like a big community. We got to discuss different collaborations between different flagship projects as well as within our own flagship projects. And there was a lot of sessions that were designed to make us think about how these collaborations could be made, what could potentially be done between us, and how groups that you would think wouldn't work together so well could still mesh and make things work. So the collaborative aspect of Tigris has always felt particularly difficult for us being outside of the social sciences. We could quite happily collaborate with the other plant scientists, but the ways in which Tigris has made us mesh with the social sciences and the archaeology and all of these different engineering type aspects of Tigris outside of the plant science seemed kind of alien to me, I think, before this event in a lot of ways. There were certain events other than this that made me think about it. For example, there was an event in Cambridge that was also a large collaborative thing hosted by Tigris, but smaller than this event, that allowed me to see how my work could interact with the archaeology on Tigris in a good way. And I got to talk to Adam Green a lot whilst I was there. But this General Assembly was the first event where I got to see all of the different FPs working together, and how everyone was meeting with each other, talking about their different projects, how their projects could mesh with other projects, and how we could take this forward. And we planned a lot of collaborative projects whilst we were here, as well as getting a lot of training that was very useful again. And sadly, not long after this, the pandemic kicked off. So whilst the collaborative projects have continued to be pushed forward, some of the more hands-on transfer type situations didn't happen. So originally, after this event, I'd planned to spend 3 months working out in ICRISAT, but sadly, that hasn't happened because of the pandemic. But hopefully, because we'll still be keeping these collaborations between our people, our PIs are still going to keep in touch, and I'm going to keep in touch with a lot of the people that I've met on Tigris. The collaboration might not have happened immediately, and the exchange might not have happened immediately, but there is still definitely room for this to happen. And I'm really hopeful that, you know, given the opportunity in the future, they will still happen. Okay, so now this is a bit of a tangent away from what I've already talked about. So everything that's happened for me with Tigris as a community really stemmed from these big events where we got to meet people, talk together, and other events like online events that we've had, like Tigris Show and Tell, where we got to continue to talk to people online and discuss our research with each other. But the events that make Tigris my own, the events that are personally mine, are my research. And I'm not going to lie to you, this is what I want to talk to you about. But I'm not going to go into too much depth. Don't worry, this isn't about to turn into a plant science talk too much. So, sorghum and pearl millet and Tigris, and what do these mean to me? So when I began working on Tigris, I'd just finished my PhD, and during my PhD, all I worked on was Arabidopsis, which, if you don't know, it's a weed. It's a research plant. It's not a crop. You don't grow Arabidopsis commercially for anything except selling it for research purposes. But sorghum and pearl millet are crops, and more than that, they're massively efficient crops. They, you know, you look at sorghum and pearl millet and you compare them to wheat, and they're way better at using water and generating biomass under poor conditions. They're way more tolerant to drought, and after a drought period, they recover much, much quicker. And all of these things make sorghum and millet really exciting. And on top of that, we're being told, okay, this is Tigris. We know sorghum and millet are great. We want to know how we can make them better. And for me, I was like, okay, great. Where do I start? I'd not worked with crops before, and my lab has never worked with sorghum and pearl millet before. So from the get-go, I'm immediately talking to our collaborators in India. I'm asking, where can I get seed? Where can I, you know, grow these to get the best conditions? What are the situations that I need to grow these in so that I can get these plants to be their best for my research? So that immediately meant that I was collaborating from day one. We were emailing backwards and forwards. I was sending them photos of my plants. So you can see in the picture on the left here, we have some sorghum plants with some slightly yellowed leaves. And that's one of the things that I sent immediately. Okay, well, I'm getting slightly yellowed leaves. Is this a problem? And in some cases, it's not a problem. But in other cases, it meant I needed to treat the plants slightly differently. So I've been able to develop what I've been doing over time. And then the picture on the right, we have some pearl millet. And this is a drought test that we're doing at the moment inside a phenotyping platform. And you can see in the foreground, we have some quite small plants that they've been droughted for 2 weeks, and then they've just been given some more water. They've had a weekend to start recovering, and they're already looking nice and healthy. So we already know these plants look great. You know, compared to what you would expect to see from similar plants put under similar conditions, these plants are doing fantastically. So why does this matter, and how are we going to be able to improve this? So sadly, I do have to do a little bit of scienciness here. So intrinsic water use efficiency is one of the things that I've really been looking at in sorghum. And this is a leaf level way of looking at how plants make use of water in order to grow better. So an efficient water using plant for us is a plant which photosynthesizes more for each piece of, or for each unit of water that it loses, or is capable of photosynthesizing quicker without losing water than another plant. So this equation here is intrinsic water use efficiency is equal to photosynthesis over stomatal conductance, which essentially means the ratio of how much carbon the plant can take from the atmosphere and put into itself and make more of itself with for each little bit of water that can escape through the pores in the leaves into the atmosphere again. And that's a large part of why plants lose water, is they take up water through their roots and it escapes through the leaves. So for a plant to be really efficient, we want it to take up water and use it, not just lose it all through the leaves. But there's other reasons to lose it through the leaves, which we'll get into a little bit. But this is what makes sorghum millet so efficient. It's this equation for us, especially on a leaf level. So what I want you to take away here is sorghum pearl millet are incredibly efficient because the ratio of carbon they take up to the water they lose is really good. It's a really strong water use efficient plant. But how do we dig into this a bit more? So how do we find things that could be improved in this? If we already know that they're great, how do we make them better? Well, we do that by looking at leaf level traits. So in sorghum millet, we've been looking at the leaf anatomy and the stomata. So what matters about leaf anatomy and what matters about stomata? So here we have, on your left, we have a sorghum plant. So just for an example here, this is a picture of a sorghum plant in the field, not strictly one of my own plants, but nonetheless a nice example with a nice picture of the vein with this little midrib that you can see. And if we zoom in on that plant, on that leaf, we then have this red picture, which is a leaf that's been stripped away and dyed so that you can see the veins nice and clearly. So the veins are all in red and the mid vein is the dark red line through the middle. And if we zoom in even further, you start to see these stomata. So these blue glowing bits on the third picture along are stomatal pores, which are these little tiny gateways on the surface of the cell that the plant, on the surface of the leaf, that are made up of a couple of cells that let the leaf open and close the, I guess, doors from the edge of the leaf to the inside of the leaf. That means that gases can't move in or out of the leaf. But why does that matter? Well, the reason that matters is that they need to open up to let carbon dioxide in. If the plants want to photosynthesize, they need to get carbon dioxide out of the air. So they need to have openings so that they can, for want of a better word, breathe in. They don't have lungs, but you know. But equally, whilst those are open, the water vapor inside the leaves, which is a higher density of water vapor inside the leaf than there is outside, is going to escape. So this is where we're getting this trade-off again. This is the water use efficiency coming from a stomatal, from a leaf level. So CO2 in, water out, stomata, these matter. And then furthermore, stomata, why do they matter? Well, there's hundreds of them. You know, this picture is a microscopic picture of, you know, a tiny area. I've not got the specific scale of this one because I cropped it down a bit so that we could see them a bit clearer. But there are hundreds and hundreds of stomata on the surface of the leaf. You know, in this example here, I have a few accessions of sorghum that we've measured that were selected because they had particularly interesting traits in the field out in India. And in these accessions, you're seeing between 50 and 100 and sometimes more than 100 stomata for every square millimeter on the surface of the leaf. So you can imagine on the scale of a sorghum leaf, which if you don't know, you know, a sorghum plant can grow easily to 2 meters tall, and the leaves are, you know, as big as my forearm sometimes. So if the leaf has this many stomata on the surface, how much, you know, what does that mean? Well, it gets slightly complicated here because it's not just the stomatal density, the number of stomata that matters, it's also the size. And it turns out that that varies a lot across sorghum and millet. And different plants have different strategies for how to optimize this. But sorghum and millet in particular have hundreds and hundreds of small, tiny stomata, which can open very, very quickly. And it's this speed, along with the number and the size, that really allows sorghum and millet to be particularly efficient, and is something that we've been looking at a lot. So in the name of the speed, I'll quickly talk to you about the speed of responses to environmental change. So this, I promise, is the last science that I'm going to talk to you about. So the two keys to speed of environmental change as far as water use efficiency is concerned, and and some of the ways that plants lose water a lot in nature is when the stomata open and close in response to, say, clouds going overhead, making the light levels change. The speed of the change in the photosynthetic biochemistry, the way that the plant chemically can take in the CO2 from the atmosphere, doesn't keep up with the speed of the stomatal movement, the speed at which the stomata can open or close to let CO2 in when they want it and close to prevent the water from escaping when it's less beneficial, when the light levels are lower. So in sorghum here, in these two graphs, we have the speed of photosynthetic biochemistry on the left-hand side and the speed of stomatal movement on the right. So before we say anything, these are really fast. I'm not showing any of the plants here, but these are very quick responses. But the thing to take away here is the red bars on these graphs indicate advanced or improved cultivars of sorghum that we've been looking at. And all the grey bars show the traditional cultivars, the land races, breeding and research material, and exotic germplasm lines. So these are the ones that aren't the really high yielding field lines. These are the ones that have potential to be used in future breeding programs to make even better plants that aren't already being used to make the ones that people would buy commercially to grow commercially. So really interestingly, some of the fastest lines, the ones with the lowest time constants, are the traditional cultivars and the land races. And some of the ones that are slower are the elite lines. And this is, fascinating. This means that the elite lines might be growing faster, but they're actually not that efficient when it comes to the speed of their responses to environmental change. So if we can improve these responses and combine them with the higher yielding qualities of these elite lines, then we still have the potential to make lines that are even more efficient as well as being high yielding. So we're digging through this data a bit more, and we're hoping to publish this quite soon. But essentially what we found is that there is huge variation there to be exploited in even the relatively small number of lines that we've been able to look at. I should also mention that all of these lines have either directly or indirectly come from ICRISAT, and we've been helped by all our partners at ICRISAT to source these from the places that they came from. So we really need to thank Jana especially for helping us get access to all the seed for this project, which, frankly, wouldn't have been possible without her help. So without further ado, that's the end of my talk. Thank you all for listening, and I really hope that you'll workshop to see me in person, ask me some questions on the day. Thank you very much.